Hey there, allow me to introduce myself. I am Charlie, the super synapse, and I live in Rebecca's brain. My job is to help Rebecca make sense of the world through her different senses. Every day, Rebecca and her mother pass by the same objects, sounds, and smells, which go through the working memory process over and over, thus committing it to long-term memory. Before something can become long-term memory, it starts out as working memory or short-term memory. In 1974, Alan Baddeley created his working model of memory to better describe the encoding process of short-term memory, which incorporates the visual spatial sketchpad, what we see, and the phonological loop, what we hear. I wonder what kind of messages I'll be sending today.
You excited for school today, honey?
Yeah.
You had a nice lamppost.
I do like the lamppost. I like the balls on top.
They are big and pretty.
Yeah.
Every time Rebecca sees an object, her eyes reflect information from the visual field. Things we see are processed in the visual spatial sketchpad in the inner eye. The visual spatial sketchpad is very important for navigation. Her two optic nerves run to the optic chiasm, which crosses over just before the pituitary, and then to the brain for processing.
You kids, you gosh darn kids, you get out of my yard. Go. Out. Shoot. Get a move on.
Look, it's Fluffy.
Out of control!
Fluffy?
Good morning, Mrs. Johnson.
Hello, dearies. How are you doing on this fine day?
Good. Wonderful. How are you?
I'm fantastic. I'm going to be baking some cookies later if you'd like some.
We would love some. Wouldn't that
That'd
be
be
nice,
great.
honey?
All right, we'll see y'all then. Bye.
Yes, honey.
I think I'm too big for you to come walk me. I think I can walk home from school on my own.
I don't know, honey. It's a long way.
No, I can remember.
Are you sure?
Yeah.
Okay.
Rebecca, where's your mom? Is she not picking you up from school today?
No, I get to walk home all by myself.
Oh my gosh, you're such a big kid.
I know, I'm excited. Wait, what street am I supposed to turn on? I don't...
The hypothalamus initiated Rebecca's sympathetic nervous system, kicking me into gear. Innervation for this first begins at the cortex of the ciliospinal center, also known as Budget Center, after German physiologist Jules Ludwig Budge. I help send postsynaptic neurons all the way to the brainstem and then finally exit through the cervical sympathetic chain and the superior cervical ganglion.
Oh no, there's Miss Johnson's cookies. I must still be going the right way.
If I can get this scent molecule to Rebecca's hippocampus, I can match it to the memory from before, and she'll remember Mrs. Johnson's cookies and be able to follow the smell home. First, I'll have to go through the olfactory epithelium and the bulb, and then the mediodorsal nucleus of the thalamus, the orbital frontal cortex, amygdala, and then I'll finally be at the hippocampus. Phew, what a journey!
Hello, dearie. How are you doing today? I baked you some cookies. Would you like one?
Oh, thank you so much.
Absolutely. Take as many as you want. Here, take the whole plate.
Okay.
Enjoy.
Oh, thank you.
Now move on. Scoot, scoot.
Charlie here! Did you know that when Rebecca tried Mrs. Jenkins' salty cookie, she was using one of the 5 taste sensations? When Rebecca tasted the cookie, she was sending a message from her taste neurons to the thalamus and into the insula, which is in the frontal lobe of your brain. When a taste stimuli is detected by a specific receptor, it is then sent to a different but specific locations in the insular cortex. This is called label line coding.
Look, there's Fluffy again.
Got out of control! I'm so sorry! Oh my goodness! Fluffy!
The things we touch are processed by neurons in the somatosensory cortex. The brain reacts to the vibrations caused by touching the object. Your skin has a bunch of different receptors. There are 4 different types of mechanoreceptors: Pacinian corpuscles for vibrations and pressures, Meissner's corpuscles for light touch, Ruffini's endings for slow vibrations, and Merkel's disc for pressure and texture.
I got it, you kids.
Oh, I'm your old man Jenkins.
Get
I must be
it
going
going,
the
get out of
right
my
way.
Go. Shoot, shoot. Never come back. You too, Missy. Get a move on. Let's go. Shoot.
Hey, Super Charlie here again. Oh man, whoever is yelling is really loud and sounds really angry. Let's help Rebecca remember who this voice belongs to. Maybe then we can help her remember how to get back home. Sounds are processed by going through the phonological loop, which consists of 2 parts: the phonological store or inner ear and the articulatory control process, or the inner voice. Inside your ear is a tympanic membrane that vibrates when a sound wave hits it. That membrane sends energy to the smallest bones in your body, the ossicles. The energy in the ossicles moves really quickly into the cochlea, where the sound is then further transformed into a neural impulse. The sound from whoever is shouting creates a reaction that shoots from the auditory nerves in the cochlea to the neurons that pass the message all the way to the brainstem. From the brainstem, the message is carried to the temporal lobes first before it moves into the auditory cortex. The auditory cortex is where a sound can trigger a memory in action.
Yay, the lamppost! I knew I'd make it home. See, Mom?
Hi, honey.
Hi.
Did you have a good day
it, school. I did.
Oh, wonderful. I'm so proud of you.
Hooray! We were able to get Rebecca back to her house, safe and sound. Your memory is definitely impressive, which is probably why it is studied a lot. Sometimes the opportunity to study it is caused by other means. One of the most famous case studies of memory is patient H.M., who recently passed away. His hippocampus was removed and therefore unable to form new memories, and also lost some of his old ones too. When he died, researchers at the University of California in San Diego sliced up his brain into over 2,000 slices in order for neurologists all over the world to be able to study it. While researchers were working on his brain, they discovered that, contrary to popular belief, a small portion of the hippocampus was actually left untouched, which led to the latest theory that the removal of the pathway from the hippocampus to the rest of the brain, the entorhinal cortex, was what truly causes memory impairment. I really hope you enjoy learning about what I do. I enjoyed teaching everybody about my job. I also hope you will take care of your brain and your body since it is doing amazing things every day.